Welcome to Biotic World. Today we are unraveling the complex and fascinating process of DNA replication in all its detailed steps. Get ready to dive into the molecular machinery that ensures faithful duplication of DNA with each cell cycles. Before we jump into the replication process, a quick recap on DNA structure. DNA is a double helix made of nucleotides, each containing a phosphate group, a sugar molecule, and one of four nitrogenous bases adenine, thymine, cytosine, or guanine. DNA replication starts with initiation, where specialized proteins and enzymes set the stage for unwinding the double helix. Our first step is the origin recognition complex. In eukaryotes, replication begins at specific sites called origin of replication. Here, the origin recognition complex (ORC) binds to DNA to identify these starting points. Following ORC binding, helicase loaders help attach MCM helicase, the enzyme responsible for unwinding the DNA strands. In prokaryotes, DNA proteins perform a similar role, binding to origin sequences and binding the DNA to prepare it for unwinding. Once helicase is loaded, it begins unwinding the DNA to create two single strands that can act as templates. This unwinding process is structure called the replication fork. First, the action of helicase. The enzyme helicase moves along the DNA, separates the DNA strand by breaking hydrogen bond between bases. This step requires energy, which helicase derives from ATP. As helicase unwinds the DNA, there is a second called as the single strand binding proteins or SSB attached to the exposed single strand, preventing them from re-annealing or forming secondary structure. Here our third enzyme scheme, the third factor, that is the topoisomerase. Unwinding creates tension ahead of the replication fork. Topoisomerase relieves this supercoiling by making temporary cuts in the DNA strand, allowing it to untwist and then rejoining the DNA. Without topoisomerase, the DNA would become overwound and hinder replication. DNA polymerase, the enzyme that synthesizes new DNA, can only add nucleotides to an existing strand. So primase, an RNA polymerase, creates a short RNA primer at the origin. This primer provides a starting point for DNA polymerase. The primer will eventually be removed and replaced with DNA, but it's crucial for starting replication. Now that primer are in place, DNA polymerase begins adding nucleotides to synthesize the new DNA strands. Polymerase third in prokaryotes, also known as the DNA polymerase delta and epsilon in eukaryotes. On the leading strand, DNA polymerase synthesizes continuously in the 5' to 3' direction. For the lagging strand, it synthesizes in short fragments called Okazaki fragments, also in the 5' to 3' direction. To keep DNA polymerase attached to the template, a sliding clamp protein encircles the DNA. In eukaryotes, this is known as the pCNA, proliferating cell nuclear antigen, and in prokaryotes, it's beta clamp. The clamp loader complex helps load the clamp onto DNA, ensuring efficient and continuous synthesis. Once the DNA is synthesized, the RNA primer needs to be removed and replaced with DNA. In eukaryotes, RNase H and FEN1 remove the RNA primers, while in prokaryotes, DNA polymerase 1 does this job, replacing the RNA nucleotides with DNA. After the primers are removed, DNA ligase seals the gaps between the Okazaki fragments on the lagging strand. Continuous strand DNA strand just got formed. Finally, we reach termination. This process differs slightly in prokaryotes and eukaryotes. Eukaryotes, termination and telomeres. In eukaryotes, termination occurs where replication forks meet or when the polymerase reaches the end of the chromosome. Telomerase, a specialized enzyme, extends the end of the linear chromosome to prevent chromosome shortening with each division. In prokaryotic termination, with circular DNA, termination sequences or TER sites bind TER proteins to stop replication forks when they meet. The completed circular DNA is then separated by the topoisomerase 4. To recap, here is a quick summary of the key players in DNA replication. Initiator proteins works like ORC in eukaryotes and DNA in prokaryotes. Helicase, it unwinds the DNA strand. Next, single strand binding proteins SSBs stabilize single stranded DNA. Next is topoisomerase, relieves supercoiling tension. Next is primase, lays down RNA primers. DNA polymerase synthesizes new strands. Next is slide and clamp loader ensures DNA polymerase remains attached. Next is RNase H and DNA polymerase 1. These remove and replace RNA primers. DNA ligase joins DNA fragments. Telomerase extends chromosomes in eukaryotes. And that's the complete story of DNA replication. This process, guided by an orchestra of specialized proteins and enzymes, ensures that each cell inherits an identical copy of DNA. Don't forget to subscribe to Biotic World more in-depth biology breakdowns and feel free to comment below with questions and suggestions for future topics.